We didn't have reagents, we didn't have blood samples from survivors, we didn't have anything, but we knew we needed to launch, so we did. Our team pulled together and started the process. When we first heard this, we thought, that's silly, that can't ever be done. This type of work takes years. I think in the last decade, we perfected a process in which we can make antibodies in about 6 to 24 months. But the more we thought about it, we thought, well, we probably can go quicker than we go now. And so with our team, we started a stopwatch, did a simulated epidemic, and we went as fast as we could, and we got the whole discovery process done in about 78 days. Of course, we had 20 years of fundamental research about the human immune response that we were using as a basis. So we knew we could speed up the process. The first known case in the United States walked into an urgent care in Seattle, Washington on January 19th. And we had to pivot, and we did the real exercise to do discovery of antibodies for COVID. The blood sample became available on a Saturday evening. We were able to contact the CEO of FedEx, who contacted his team, and were able to move the sample with a GPS tracker overnight into Memphis. One of the company folks drove in a Lincoln Town Car to my home in Nashville, Tennessee, and handed me on a Sunday morning the sample, and I rushed off to work. So the sample was actually about 2 days old by the time we got it, and we were so excited to get it into the lab and start working. But when we looked at the cells under the microscope, they were not alive. And then we started brainstorming a little bit, and we realized that the genes for the antibodies that we were seeking were still in the tube. So we sequenced the RNA and DNA that was in the blood tube and made a library to decode the recipe for all of the antibodies there. We needed to move so quickly in this instance that we could not rely on our usual techniques. So we turned to next generation sequencing, supercomputing and bioinformatics and artificial intelligence, and single cell biology, in which we were testing individual cells from human bodies one at a time, but doing that in thousands. And we had to do this work under very uncertain circumstances. Many of the scientists were working as many as 20 hours a day. Once we identify sequences that are interesting to us, that's just letters on a computer. We need to physically make the DNA, and we use instruments that can synthesize the DNA. It's like a desktop printer. We put in a code and DNA comes out that we can use to then make antibodies. So we obtained blood samples in March, and about 24 days later, we were able to pass on the recipe for the cure, the DNA sequences of antibodies that we discovered, to a pharmaceutical partner who could do the manufacturing. Of course, we want to do these things quickly, but also safely. So they were testing the materials they had manufactured for several months, working with the FDA to ensure they were safe. And by August, they were using the antibodies that we had started making in March. So that period was very, very compressed and rapid, but still we went through all the required steps to be safe. And now we're into what are called the phase 3 trials, which are the efficacy trials to see how well these molecules will work to prevent and treat the infection. Often people wonder, haven't we exhausted the number of studies that we can do on something like the immune system? I think the more we learn, we sort of are just chipping away at the edges, and we see there's so much we don't understand. Medicine, therapeutics, prevention, vaccines, they all grow out of the curiosity of how the world works, which is discovered through basic science.